Good morning, everyone, and welcome. I am Dr. S.K. Singh, and today I am excited to talk to you about the qualification of autoclaves, specifically steam sterilizers. This is the first slide of our presentation, and I'll be guiding you through the key aspects of this important topic. Over the next few minutes, we'll explore how autoclaves work, their significance in sterilization, and the standards that ensure their effective operation. So, let's get started and dive into the world of steam sterilizers. Let's start by understanding what an autoclave is. Essentially, it's a steam sterilizer that uses pressurized steam to kill microbes on various items such as instruments, glassware, and media. The typical set points are around 121 degrees C at roughly 15 psi pressure, and the sterilization cycle usually lasts between 15 to 30 minutes, depending on the load type. These devices are widely used across different fields like pharmaceutical, clinical, research, and quality control laboratories. Now, you might wonder why qualification of this equipment is so important. Well, regulatory bodies expect us to have documented evidence that our autoclaves are functioning reliably. This is part of the broader framework of ICH Q9, which focuses on quality risk management. Ensuring our autoclaves are properly qualified helps us meet these standards and ensures the safety and efficacy of our processes. So, in essence, understanding what an autoclave does and why qualification matters is fundamental to maintaining compliance and safeguarding product quality throughout our operations. Let's take a closer look at the qualification lifecycle overview. This process is composed of 3 key qualification stages that are essential for ensuring the autoclave functions properly and reliably. First, we have the IQ, which stands for Installation Qualification. This stage confirms that the autoclave has been installed correctly according to all specifications and requirements. Next is the OQ, or Operational Qualification, where we verify that the autoclave operates within the set parameters under normal operating conditions. Finally, we have the PQ, Performance Qualification. This phase assesses whether the autoclave can consistently perform with actual loads, ensuring it meets the necessary performance standards over time. These 3 stages follow a sequence: IQ, then OQ, and finally PQ, and together they demonstrate that the autoclave is installed properly, operates correctly, and delivers consistent results. This structured approach is crucial for quality assurance and compliance in our sterilization processes. Let's talk about Installation Qualification, or IQ. The main goal here is to make sure that everything has been installed correctly and that all the documentation is in place before we move on to functional testing. This step is crucial because it sets the foundation for everything that follows. First, we want to verify that the equipment matches the purchase specifications. This means double-checking that we've received the right equipment and that it conforms to what was ordered. It's about ensuring accuracy from the start. Next, we need to confirm that utilities like steam lines, power supplies, and water feeds are properly connected. Proper connections are vital because they impact the equipment's performance and safety. If these aren't set up correctly, it could lead to issues down the line. We also check for calibration certificates for critical instruments like thermometers, pressure gauges, and data loggers. Having valid calibration certificates ensures that measurements are accurate and reliable, which is essential for maintaining quality and compliance. Finally, safety and alarm systems must be installed according to the original design. These systems are our safety nets. They alert us to potential problems before they become serious. Ensuring they're correctly installed is key to creating a safe working environment. Overall, IQ helps us confirm that everything is in its proper place and ready to support reliable, safe, and compliant operation as we progress through the project. Let's take a closer look at Operational Qualification, or OQ, which is a crucial step in ensuring our autoclave functions as intended. The main goal here is to demonstrate that the autoclave operates within its specified design parameters. This phase really focuses on controlling key parameters under different conditions, both when the chamber is empty and when it's in use. Now, some of the typical tests we perform during OQ include the vacuum leak test. This test is essential because it confirms that the chamber can hold a vacuum without any significant leaks, which is vital for effective sterilization. Another important test is the Bowie-Dick test. This routine daily check verifies that the autoclave can properly remove air from the chamber, especially in pre-vacuum models, ensuring that sterilization conditions are uniform throughout. We also conduct an empty chamber heat distribution test. This involves temperature mapping at various points inside the chamber to make sure heat is evenly distributed, a key factor for effective sterilization. Lastly, we check the pressure and alarm functions. This step confirms that the pressure controls are working correctly and that alarms will activate properly if any parameters go outside of safe limits. All these tests together help us verify that the autoclave is operating reliably and safely, which is fundamental to maintaining high standards in our sterilization processes and overall quality assurance. Now, let's take a closer look at this OQ heat distribution example within an empty chamber. The set point we're aiming for is 121 degrees Celsius, with a tolerance of plus or minus 2 degrees. This ensures we maintain consistent and accurate temperature control. To monitor how evenly the heat is distributed, we place between 10 and 16 calibrated thermocouples around the chamber. It's important to position these sensors thoughtfully, around the corners and the center, to get a comprehensive picture of the temperature profile. By doing this, we can identify any hot spots or cold spots, which is crucial for ensuring uniform heat distribution. This example highlights the importance of thorough sensor placement and calibration in achieving reliable temperature control, a key theme in our overall process. Maintaining this balance helps us optimize performance and ensure the quality of end products, especially in sensitive applications. Let's take a look at this example involving a vacuum leak test, specifically the Bowie-Dick test. The key point here is the maximum pressure rise during the hold period, which should be less than or equal to 1 millimeter of mercury per minute. This measurement is crucial because it helps us determine whether there's a leak in the vacuum system. If the pressure rises faster than that, it indicates a potential leak that needs to be addressed. Ensuring this parameter is within limits is essential for validating the integrity of our sterilizer's vacuum cycle. Remember, maintaining this standard helps us ensure that our sterilization process is effective and safe. This example ties into the overall theme of quality assurance and the importance of routine testing to prevent issues before they impact patient safety. Let's talk about performance qualification, or PQ. The main goal here is to demonstrate that our sterilization process consistently performs as expected under routine operating conditions, and this is tested using actual loads. When we conduct PQ, we're looking at several key components. First, we need to assess the heat distribution and penetration within the loaded chamber. This ensures that every part of the load receives the proper sterilization conditions. Next, we use biological indicator challenge tests, typically with spores like Geobacillus stereothermophilus, which are highly resistant, usually at least 10 to the 6th spores, to verify sterilization efficacy. Placement of thermocouples is also critical. We position them at cold spot locations, areas that tend to be the most challenging to sterilize, to make sure the process is effective throughout the chamber. And finally, we perform at least 3 consecutive cycles for each load pattern to confirm consistency and reliability over time. All of these steps work together to validate that our sterilization process is robust and dependable, which is essential for maintaining safety and compliance in our operations. Let's take a closer look at the PQ example test plan. First, we have the load types, which include porous loads, glassware, and media bottles. These are representative items that we typically sterilize, and it's important to test these to ensure our sterilization process is effective across different materials. Next, we have the sensors and biological indicators. We've placed 10 thermocouples at critical points throughout the chamber to monitor temperature accurately during the cycle. Additionally, there are 10 biological indicators positioned right next to those sensors, providing a direct measure of sterilization efficacy. Moving on to the acceptance criteria for this performance qualification, or PQ. The key points here are that the chamber temperature must stay between 121 and 123 degrees Celsius during the hold phase. Maintaining this temperature range is crucial for effective sterilization. We also need to ensure there are no cold spots within the chamber, which could compromise the sterilization process. Lastly, all biological indicators should show no growth after incubation, confirming that the sterilization cycle successfully eliminated all microbial life. Overall, this test plan helps us verify that our sterilization process is reliable, consistent, and effective for the loads we typically process. Now, let's take a closer look at the lethality calculation using the F0 concept. The F0 value is essentially a measure of sterilization lethality at 121 degrees Celsius, expressed as a time equivalent. When we have an actual process time above 121 degrees C, say, 18 minutes with proper steam penetration, we typically aim for an F0 of at least 15 minutes, which is often considered our minimum target. The key point here is that we achieve the lethality target when the accumulated F0 equals or exceeds the required value for sterilization. Moving on to the PQ results and a calculation example, first, the temperature spread calculation. The maximum measured temperature was 122, 8 degrees C, and the minimum was 121, 2 degrees C, giving us a spread of 6 degrees C. Because this is within the acceptable range of plus or minus 2 degrees C, we pass this criterion. Next, the biological indicator results. All 10 biological indicators showed no growth after incubation, which confirms that sterility has been successfully achieved. Finally, there's a graph showing temperature versus time during the hold period. This visual helps us verify that the process maintained the necessary conditions for the required duration, ensuring effective sterilization. In summary, these calculations and results collectively confirm that the sterilization process met both the temperature and biological criteria for effective sterilization. Let's take a closer look at the key documents involved in this process. First, we have the IQ protocol and report, which ensure that all equipment and systems are installed correctly and meet the required specifications. Then, the OQ protocol and report focus on verifying that the equipment operates as intended under controlled conditions. The PQ protocol and report go a step further, confirming that the process consistently produces the desired quality in real-world production settings. Additionally, calibration certificates for sensors are essential to demonstrate that our measurement devices are accurate and reliable. Raw data logs, including temperature and pressure readings, are maintained to provide traceability and support compliance. Biological indicator results are also critical, as they verify sterilization effectiveness through biological testing. From a regulatory perspective, it's vital that we follow controlled written protocols with clearly defined acceptance criteria established before testing begins. This helps ensure consistency and compliance throughout the process. Now, regarding documentation and GMP compliance, it's important to develop and adhere to SOPs for key activities. These include operating the autoclave, calibrating sensors and instruments, performing charger or BOWI, DIC tests, and managing load configuration and biological indicator placement. Each SOP ensures that procedures are standardized and consistently followed. Finally, when it comes to change control, requalification becomes necessary after any major maintenance, equipment relocation, or significant load changes. This ensures that the sterilization process remains validated and reliable, maintaining the overall integrity of our sterilization program. Now, let's look at some key mitigation actions that can really make a difference. First, adjusting the steam supply and the air removal mechanism helps ensure proper sterilization conditions and prevents issues caused by inadequate steam or air pockets. Improving gasket sealing is also crucial, because a good seal prevents leaks and maintains the integrity of the sterilizer environment. Lastly, rebalance the load pattern and retest. This step ensures that everything is evenly distributed, which helps achieve consistent sterilization results. When we talk about common failures, it's important to recognize these typical issues. Uneven temperature distribution can compromise sterilization, so identifying and fixing it is essential. A failed Bowie-Dick test is a red flag indicating that the sterilizer isn't effectively removing air, which can hinder sterilization. A positive Bowie test suggests contamination, and a leak rate above threshold indicates potential breaches in the sterilizer's integrity. Addressing these failures with the right mitigation actions is critical to maintaining the safety and effectiveness of our sterilization processes. This connects directly to our overall goal of ensuring reliable sterilization outcomes and patient safety. So, in conclusion, let's reflect on the key points we've discussed. First, qualification is essential because it assures us of sterilization performance. When we qualify a system, we're confirming that it consistently meets the required standards. Next, IQ, or installation qualification, helps us verify that everything has been installed correctly. This is fundamental to ensuring the system functions as intended right from the start. Moving on, OQ, or operational qualification, takes us a step further by assuring that the equipment performs within established limits during normal operation. This is crucial for maintaining consistent quality. Then, PQ, or performance qualification, demonstrates that the sterilization process works effectively under real-world conditions, with actual loads, replicating everyday use. And finally, documentation plays a vital role. It's not just paperwork, but a critical component for GMP compliance and inspections. Proper documentation ensures transparency, traceability, and that we meet regulatory expectations. Altogether, these steps form a comprehensive framework that guarantees our sterilization process is reliable, compliant, and effective from start to finish. For further support, please feel free to contact us. We're here to help you with any questions or additional assistance you might need moving forward. And with that, I want to sincerely thank you all for your time and attention today. Your engagement is truly appreciated, and I hope you found this presentation valuable. Remember, we're just a contact away if you need anything more. Thank you again.